Plants store the energy that they create in a variety of different forms. Sometimes it's sweet apples. Other times it's crunchy and crisp carrots. Sometimes it's in the seeds of grain crops that we grow. This lesson is on the light-independent reactions. So once enough ATP, adenosine triphosphate, and NADPH have been produced by the chloroplasts, glucose can then be synthesized. So this involves a series of reactions known as a Calvin-Benson cycle. And these are the light-independent reactions because they don't need light energy directly to take place. The Calvin cycle regenerates its starting material after molecules enter and then leave the cycle. So all the material that is needed for the Calvin cycle is present in the cell and is reused. So carbon dioxide enters the cycle, but it leaves as part of the sugar molecules. And the cycle spends the energy of ATP and the reducing power of electrons carried by NADPH to make the sugar. So all those electrons that ATP and NADPH gained are used to make glucose molecules. The actual sugar product of the Calvin cycle is not directly glucose, but it's a 3-carbon sugar known as glyceraldehyde 3-phosphate, or G3P. And then from here, different plants will make different types of sugar, starch, or cellulose products. So we'll talk about G3P more often in the Calvin-Benson cycle as opposed to sugar. So each turn of the Calvin cycle fixes one carbon molecule. So for each synthesis of the G3P molecule, the cycle must take place 3 times, fixing 3 molecules of carbon dioxide. So to make one glucose, which is C6H12O6, the Calvin-Benson cycle would need 6 cycles and 2 G3P molecules, fixing 6 molecules of carbon dioxide. So the Calvin cycle has 3 phases. Phase 1 is carbon fixation. So in the carbon fixation phase, each carbon dioxide molecule is attached to a 5-carbon sugar. So here it says input 3 carbon dioxide. So we're just going to go through the cycle once, knowing that the cycle would have to go through 3 times to make one G3P molecule. So the carbon dioxide molecule is attached to a 5-carbon sugar known as ribulose biphosphate, or RUBP. So this is catalyzed by RUBP's carboxylase, or rubisco. The 6-carbon intermediate splits in half to form 2 molecules of 3-phosphoglycerate per CO2. So we have 2 molecules of 3-phosphoglycerate per carbon dioxide molecule that's been fixed in this first step of the Calvin cycle. Our second phase is the reduction phase. So our 3-phosphoglycerate receives another phosphate group from ATP to form 1,3-biphosphoglycerate. So we have gained electrons, so we are being reduced. A pair of electrons from the NADPH reduces each 1,3-biphosphoglycerate to G3P. So this is 1 G3P would be half of a glucose molecule. The electrons reduce a carboxyl group to a carbonyl group. And then we have our regeneration phase. So this is where the carbon dioxide acceptor, RUBP, are rearranged to be able to accept a new carbon dioxide molecule from outside the system. So the cycle must spend 3 more molecules of ATP to complete the cycle and prepare for the next round of carbon fixation. For the net synthesis of 1 G3P molecule, which again would be half a glucose molecule, the Calvin cycle consumes 9 ATP and 6 NADPH. So it costs 3 ATP and 2 NADPH per carbon dioxide. And again, there's 3 carbon dioxide per G3P molecule. The G3P from the Calvin cycle is the starting material for metabolic pathways that synthesize other organic compounds, including glucose and other carbohydrates. So if we were going to make a glucose molecule, we would need 18 ATP, and 12 NADPH to make one glucose molecule. So that is a lesson on the light-independent or Calvin-Benson cycle of photosynthesis. Thanks for joining. Take care. Bye.
If you enjoyed this video, check out our website, online.plrd.ab.ca, and visit us on Facebook, Prairieland Online Academy. You can find out all that's going on with Prairieland Online Academy.